Good evening, everybody. We'll be starting our today's session, and our today's resource person is Dr. Madhavi Tripathi. Let me introduce her in brief. Dr. Madhavi Tripathi obtained her PhD from IISc Bangalore from the Department of Materials Engineering and integrated masters from Indian Institute of Information in Biomedical Engineering, Allahabad. She is currently working as postdoctoral researcher at Johns Hopkins University, United States. She has over 20 publications in national, international journals and conference proceedings. We welcome you, madam.
Thank you.
Okay, we'll be starting the today's session now, and the title of the session is recent trends in nanomaterials. Over to you, madam.
Thank you. Thank you so much. Good evening, everybody. I can see almost many people have joined, so I think we should start. So as I have she has already introduced, I'm Madhvi and I'm currently working in Johns Hopkins University. I have extensively worked in field of nanomaterials for like around more than 8 years or 9 years, and currently my work is more clinical in terms of research. So I will be sharing some of my previous works as well. So I'll start. I'll share my PPT. Is it visible?
Yes, ma'am.
Yes, madam, it is
Okay,
visible.
thank you. Okay,
And
thank
I
you.
request all the participants to ask the questions at the end of the session. Thank you.
Yeah, that would be great. Thank you. Just slide share. Okay, so good evening, everybody. It's really a pleasure to be, the part of this program. So I'm here to share my insights on the topic, as I have been working on this in this area for a pretty long time. And thankfully, I have worked on some of the, the advancing projects in the nanomaterials. So when we talk about nanomaterials, these are, these are very tiny objects, but basically they are very, very powerful. They are actually revolutionizing how we approach the problems in the industries and basically in terms of biomedical engineering. So just diving, into that, just before that, I would just let you know that this is like my affiliations, as she has already told in the introduction. So moving on. So here are the contents. This is what you can expect in the coming slides. So we will be discussing a very brief about what exactly nanomaterials are, and then how these are used in the biomedical engineering in the current scenario, and also what are the recent trends which are going on in the nanomaterials, because people have been using the nanomaterials for decades. So what exactly is going on right now? And also my work basically in my PhD, I use nanomaterials for the purpose of phototheranostics. That is, it was diagnosis and therapy using the laser light for cancer. And then we'll discuss the challenges in the future directions. You can write your questions in the comment section, and I will just request the moderator if they can keep an eye on all the questions. And in the end, we'll try to take up most of the questions. So starting with what exactly are the nanomaterials? This is a very basic concept, but still, it's important that these are the particles, these are the materials which are basically in the range of, if we go by the definition, they are in the range of 1 to 100 nanometer. But if you might have seen that even the particles more than 200 nanometers or less than 1000 nanometers, they are also considered as nanomaterials. So why is that so? Because they are not falling into definition. The thing is that, when we go by the definition, that is 1 to 100 nanometer, most of the properties, the best properties of the materials which we expect to be obtained in a nanostructure, that we get. As we move on by increasing the size, or we decrease the size, the properties which we expect from a nanomaterial actually varies. But still, in the range of 1 nanometer to 1,000 nanometer, we consider it as nanomaterials. Also, why these are so important? What makes them so unique? It's actually their quantum confinement effect, or the quantum properties which they actually show. So this range has this range of particles, this size. They have larger surface to volume ratio. So this makes these nanomaterial really highly reactive and very much efficient as compared to their bulk counterparts. And when we talk that it should not be very much smaller than 1 nanometer, why? Because at that sub-nanometer or that atomic scale, materials, they often lose their structural ability and their functional ability. So it's that's why the definition is constrained till 1 nanometer. So that's the phenomena. So the quantum confinement effect, when we talk about, this is the area where the quantum effect is observed, which is very high at that in that particular zone. So what happens in quantum confinement effect is that the electrons are confined in a very small space, which results into a discrete, it results into discrete energy levels instead of a continuous band of energies. So this discrete energy levels of electrons, it provides various properties to the nanostructures. So in the figure, you can see there are two ways to prepare the nanostructures. One is the top-down approach, and another is the bottom-up approach. In the top-down approach, we take the bulk material and we keep fragmenting it till we reach the nanoscale. And in the bottom-up approach, we go by the atoms, we take the molecules, we prepare the clusters, and then we go to the nanoscale. So the design properties, talking about the design properties, as I've already discussed in the previous slides, these are the properties, the quantum effect, the quantum confinement effect, which actually provides them with the properties such as electrical conductivity, where you might have heard of some of the nanomaterials are very good at electrical conductivity, and also they show good magnetic responses. You can see in the figure, we can see that there is a gold nanoparticle. It's showing multiple colors. So these colors are just coming because of the different sizes. And this size difference is not very much. It's just 10 nanometers, 20 nanometers, 30 nanometers, and it imparts these beautiful colors to the nanomaterials. The bulk gold looks golden, but when we go to the nano scale, the properties, they completely change. So, by changing the size and the shape and the composition of the nanomaterials, basically we fine tune the properties for the specific needs. Whatever needs we require, we might have optical requirements, for sensing applications or for imaging applications. We might have electrical requirements. We might want to harness the electrical properties at the sub-micro level range, and then we want the magnetic properties. So we can just tailor these size and the shape and how they look, and we can fine tune these properties. So these adjustable properties is what actually makes these nanoparticles quite versatile, and they are being used in the industries and everywhere, to develop sensors, catalysts, drug carriers, and many other things. So moving on, as I just discussed in my previous slide, the applications of the nanomaterials, if we see broadly, we can define, they are applied in biomedical industry, for diagnostic imaging, tissue engineering. Environmental applications is one of the areas where, these nanomaterials are being applied, for the water filtration, or, the contaminants of water currently, either it's the seawater or the sewer water, which we are using. Many people are coming up with great ideas and new novel ideas for the water filtration system, also for the killing the bacteria and purification of the water. Then in the electronics, nanomaterials are the integral part. We do like nanofabrication for the small chips and everything which are being used in our electronic devices. There it's playing a big role. And then the solar cells, if we talk about the solar cells, and the cosmetic industries, all the products which we use, ranging from sunscreen to our foundation, everything has got nanomaterials inside it. So these, so when we talk about sunscreen, just an example, just because we all are aware of the sunscreen which we use. So the sunscreen has got the zinc oxide nanoparticle inside it. And these particles, they protect and block the harmful UV rays. And it protects our skin. So unlike the traditional formulas which we used to use in the older times. So when we talk about it conclusively, we can say that, from healthcare to the environmental solutions, nanomaterials have found their way and they're creeped inside, almost all the industries, and they are there in nearly every aspect of our life. So it's important to address the beauty of these nanoparticles, and also the challenges which we face while we work with the nanoparticles during the applications of these nanoparticles in various fields. So in my talk, I'll be basically focusing on the biomedical engineering aspect because that's my background. I've done my PhD in materials engineering, so that gave me a very broad view of materials also, the kind of materials and the metals and ceramics and so many things which can be used to make the nanoparticles. So first, talking about the biomedical engineering aspect, there's a complete timeline which we can follow, ranging from 90s, from the 1960s and 1950s until now. There have been development. There are several nanoparticles which are already in the clinical trials, and there are several which could never make into it, no matter what we try. You guys might have heard something of curcumin. Curcumin, we have in India, we have done extensive work on curcumin, but it could never make to the clinical trials because there were several problems associated with it. Probably we could never deal with those problems, and it could not. And there can be other reasons as well. But overall, there are hundreds of particles which are being worked on in the laboratories, in our institutes, but they definitely do not make into the market. So but this field is, it merges the biology part, the medicine part, the engineering part to create the innovative solutions for the healthcare challenges. So here, the nanotechnology in the biomedical engineering plays a very, very crucial role. Although it's interdisciplinary, but this is a very, very important role being played by these nanomaterials. So, if we for an example, the nanoscale particles, they also align with the size of the biological molecules which we have, like proteins, DNA, and virus. So what it does, it allows a direct interaction of these nanomaterials with these very miniature molecular parts of, the medical aspect. So, so that we can, deal with, the tweaking of these things at such molecular level. And also this scale enables us to go over the traditional technologies which we could not achieve in the previous periods. We can achieve them, by targeting the disease and being precise and being personalized. So that's what it actually provides us. And also, many groundbreaking researchers are being done these days. And one of the researchers, like in John Hopkins, also recently I came across. And there are several personalized medical interventions which people are trying to do, so that if suppose we have somebody has a cancer of some specific type, how can we actually analyze the type of that cancer? Because the cancers are so dynamic and everything is like, so unique in its aspect. So these nanoparticles, the nanorobots, they help us to go in that environment and give us certain information, the functional information, the metabolic information, so that we can understand the prognosis and we can diagnose that disease very well. So that's why they have made their place in the biomedical industry. So these are why, so I have already discussed why the biomedical needs the nano. So because of their superior imaging, like abilities. So my work in my PhD, I would just discuss that I worked on a particle system, which was actually able to do the therapy and the imaging together, which is the combination therapy. And the kind of imaging I worked on is a very new modality known as photoacoustic imaging. So later on in the coming slides, I will discuss about that. But why I'm telling that is because these particles not only can be used for therapy, which people have been doing for years, to make for the chemotherapy and for other kind of therapies. Simultaneously, a single particle can even do a therapy and side by side at the same time simultaneously can be used for the diagnostic purposes. So it gives an upper hand to the doctors. If you imagine it, suppose it comes in the clinics and it gives an upper hand to the doctor that, while performing the cancer surgery, they can image it, and side by side they can keep doing the surgery, which is currently not very much possible when we talk in terms of MRI or PET scan or SPECT scan, which is used for the cancer diagnosis. So what these nano actually provides, they have the cell to cell level interactions, as I told, in terms of when we cater with the proteins or viruses and other things, and then they provide a controlled release. In the conventional drug type, what happened that when we used to give the drug either by IV or by in the form of tablet, it goes everywhere, and either it gets eliminated, it never goes to a particular targeted place, and also there is not a controlled release. There's always a burst release. Immediately there'll be a lot of release, and then slowly over the period of time it subsides. So we want for something to cater a problem like cancer, we really want a controlled release over a period of time, and there should be a bioavailability of the drug in the blood. But in the current scenario, the conventional these drugs, they generally get eliminated outside the body. And also when we talk about the tissue engineering, so tissue engineering, you might have heard, the artificial organs. Now people are creating artificial organs so that, if suppose we have a hand cut or some organ is, not working in our body, we can actually use our cells and tissue engineer it and create a new organ and place it there. Also, there are several scaffolds where these nanoparticles, they play a very important role in interaction with the body. When they are placed inside that like that scaffold, they play a very important role, in interacting with the proteins of the body, in interacting with the other tissues of the body where it is placed. So yeah, this is how they are just making their place inside the biomedical industry, and currently people are harnessing, exploring them like too much. So yeah, this is one aspect which I really like about these nanoparticles. So if you look at this slide, this is the periodic table which we all are very much familiar with. So if you see that these are the elements which have now been used and explored by the people to create the nanoparticles for various different purposes, various purposes. These particles are made. Several of these elements are actually present in our body, and many of them, they are also not present in the body, but they are not very much harmful also. So they are being used for the purposes like, so if you say like elements like iron or calcium or magnesium, and like zinc also, it plays a very critical role in maintaining our the processes inside the body. So they are inside the body. Iron is essential for the oxygen transport. The calcium is very much important for our bone health and our muscle functions. So when we incorporate these nanoparticles in the medical treatments, such like as dietary supplements or injectable formulas, then they give a good effect to the body, and they are in coherence with the homeostasis of the body, and things work well. Also, there are several non-essential elements in the medicine. Non-essential, when I say, that means they are not found inside our body, but they are being used for creating nanoparticles for the application in biomedical industry, like gold and silver. Gold and silver have been used extensively for their antimicrobial applications. So many, and like platinum is being used for so many chemotherapy drugs. The platinum is being used. Similarly, the radioisotopes, when we talk about the radioisotope, we would have never thought many decades before that even the radionuclides can help us to know something of a living body. But right now we can use them to diagnose the disease such as cancer and heart conditions like technetium or like iodine 131. So yeah, they are being used and they are being engineered in a way so that we can harness the best out of them. So Then moving on, we have the nanoparticles for the drug delivery. That's the work which I have done over the period of years. So why we need nanoparticles for drug delivery? So conventional drug, they don't only face the problem of bioavailability, that they are not biologically available in the blood after some time. They also face the problem of drug resistance, which is a very big problem. These the conventional drugs, when they are used for a particular disease several times, then the disease, our body mutates in certain way. The cancer cells, they mutate in certain way that they cause a drug resistance, and then the drug is no more effective for that particular disease. So what nanoparticles does, we engineer the nanoparticles in such a way that we deliver the drugs directly to the disease cells, and we try to minimize the off-target effects. And this approach ensures that the therapeutic agents, they reach the target site in the desired concentration. So we can maintain the precision, we can maintain, how precisely we can target the things, and also we can modify these nanoparticles. We can add certain ligands over it. Ligands, as in, you can understand, some people who might not know, it's like a lock-in key. Suppose you add a key kind of thing on your nanoparticle, and it goes to a particular place where only that lock is present, so it will only go and it will only be applied into that particular area. So for that, it's that also reduces the toxicity, because it's not going to the normal cells and not destroying the normal cells of the body, where it is actually not very much used. So what nanoparticles offer, they offer control delivery, precision targeting, versatile. It's a very much versatile platform, because they can be engineered from polymers, they can be engineered from lipids, they can be engineered from inorganic metals, they can be engineered from basically so many things, and natural polymers and the synthetic polymers. So yeah, we just have to keep an eye that it should not be very much toxic in the long term. That's a very basic criteria which we should involve. Most of the time when we do the research and we try to create a project, we just see the toxicity for a very lesser period of time. In the in vitro conditions, we see 24 hours or 48 hours, and in vivo conditions, just 1 month or 2 months. But it happens that, sometimes this toxicity of these nanomaterials, it only comes up and we can only see it after a very long time. So yeah, that is a very important phenomenon. That is the biggest reason, one of the biggest reason why these nanoparticles are actually not getting converted into the clinical translations. So also these nanoparticles, they have their applications in the biosensors. So they're like materials like gold nanoparticles, carbon nanotubes. They amplify the signals. So suppose there is a signal, they will just amplify it, which enables us to detect something which is in a very less concentration, like some biomarker inside our body, like some protein inside our body, which is just released on a particular condition. At those things, we can do using the biosensors. Because they have a beautiful property of amplifying the signals. The gold also does that, and there are many semiconductor particles they also do this. Also, as I've already mentioned, for the rapid and early detection. So what happens that when we talk about cancer, one very important aspect of cancer is that most of the time we get to know about the cancer in a very later stages, where the surgery is not possible. And if the surgery has been done, there is something left over, or it's a very later stage, third stage or fourth stage, nothing much can be done. So what if we can detect the disease at a very, very early state, and there only the surgical intervention or some chemotherapy might completely eradicate the disease. So for that purposes, also in terms of not just in cancer, also in terms of infectious disease, if we could actually do that, we can actually eradicate the disease before it actually goes out of our hand. So it's the same thing. And also for the minimally invasive approach, because we can do the imaging from above the body. We don't have to do the biopsies and other things. And, there are so many discomforts which a patient goes through when we take the blood and so many other things. So, yeah, these nanoparticles, the basic thing is that, no matter what we try to do with these nanoparticles, there are so many applications we are trying to figure out, but they do fail. They do fail because, when we create our projects, we do not keep in mind, the toxicity and, the long-term impact and, the bottlenecks, which are actually preventing these from getting translated. We do not keep that in the mind. And so many of the times our projects actually fail. So, yeah, these are, and also just in terms of biosensors and diagnosis, one very great example is the pregnancy tests. So in the pregnancy, there are so many pregnancy test kits right now out in the market where they actually use the gold nanoparticles. So yeah, it's a great example to understand how these nanoparticles are actually, they have already made their place into the industries very well. So talking about the main topic, which is like the recent trends in the nanomaterials. So these are the, whatever I spoke till now, these are the things which are already in the market and they are, people are already using it and, it's already there. But there are several things, which are currently being explored and hopefully they might be able to, make their place. So when we talk about the nanomaterials, one very important aspect is, we cannot forget the nanomaterial word actually comes from the biomaterial. These are the biomaterials which we can use for the medical applications, and we convert these biomaterials to basically nano size, and then we use it. So these biomaterials, they actually create the cornerstone of the modern medicine for now. So for we even if we want to go towards the personalized medicine, which is a medicine which can cater a particular patient, a particular type of disease which is unique to that patient, for that purpose also we need to have a very good understanding of biomaterials. And also when we take the biomaterials to the nanoscale, how the properties change and how can we play around with it, that is something very much important. So first, implementation. Tissue engineering is not like a very old field. I think it's just been a decade or two where people have started, exploring tissue engineering like anything. So in the tissue engineering, it leverages the nanotechnology to create, the innovative solutions for regeneration of the tissues or repairing the tissues. So if we say that, the scaffolds, they can actually, the scaffolds, they can mimic the extracellular matrix, or they can provide the framework for the cell adhesion and growth. So now, how nanoparticle and the tissue engineering, for example, suppose we can see the electrospun nanofibers and nanocomposites. So we can use the electrospun nanofibers and nanocomposites hydrogel and then create a scaffold, and that can promote actually the tissue regeneration in certain cases. Then also, for the controlled release of the growth factors, the scaffold, they can actually deliver the bioactive molecules like cytokinins, in a very controlled manner where it is actually required. So it also supports, the tissue repair and the healing. So, and in the regenerative medicine, we basically talk about the cartilage and the bone repair and the regeneration. So they're also in the form of grafts or patches, they are being used for that purpose. So, if we see that, earlier on the nanoparticle, when this field actually started, you all must be knowing that when people never thought that it can be used for tissue engineering, basically what we started was the drug delivery and the delivery of drugs for various elements like infectious disease, antibacterial things, and then for cancer. But nobody actually thought that it can actually be used for tissue engineering. Also, nobody thought that it can be used for diagnosis, which we will go ahead and we will just, yesterday, if you might have heard in Dr. Zenia's talk, that also she used the nanoparticles for, for MRI. So yeah, they are doing a pretty good job. And Yeah, we can go ahead with these particles. I really like these particles, so I can just keep talking about them. So, okay, coming to the another application, another new applications, this is known as the nanoporous membranes. So, now what nanoporous membranes are? They are materials with precisely engineered pores. So, it's like you can imagine a sheet, and they have the pores inside them, and these pores are like in a nano range, and they offer the solutions for filtration processes. And not just in the medicine you can UV, it is being used for dialysis and all. It is also being used for other areas, so for other liquids and gases. So in when we talk about in the biomedical aspect, then they are being used for to removing the toxins from the blood, and also advancing the drug delivery system for separating the specific molecule based upon the size and the properties of these. Also in the artificial organs, when we are talking about tissue engineering also, nanoporous membranes are being used to create a bioartificial kidney, because we know that the kidney does that work of removing the toxins from our body and separating the ions. So yeah, there also it is being used, and it has been shown in some of the papers. It has been shown improving the efficiency and biocompatibility. So this has the advantage over the traditional membrane. It offers like um, you know efficient, improved selectivity and efficiency, and it has reduced the fouling due to their highly controlled pore dimensions. So that's one thing which very remarkable about this process. Then coming to the smart nanomaterials. So these are the nanomaterials which in past decade people have been like exploring it like from every aspect. So what exactly are the smart nanomaterials? These are the engineered nanomaterial which respond to the specific stimuli like pH, like temperature, like light, like enzymes, and it makes them highly versatile for medical and technological applications. Why? Because we have temperature, pH, light, and enzymes almost everywhere inside our body, and even outside our body. But when we talk about biomedical applications, pH, temperature, enzyme, everything works. And even when we talk about light, we can give the light from outside, and then they can um, act in a certain particular manner. So why are these known as smart materials? So these are the materials which are actually designated to change their properties or behavior in response to the external triggers, and it enables a precise control over their function. We can actually control the way they work inside our body. So like on-demand drug delivery, and this on-demand drug delivery minimizes the side effects and maximizes the therapeutic efficiency. Then like suppose some tumor, we know that the tumor environment, the cancer environment is actually acidic in nature. So the drug will only be released only when it reaches an acidic area. Our entire body is not that acidic, but apart from some organs like stomach and all, but the cancer area is quite acidic, and there these smart nanomaterials, they release the drug under that acidic conditions. And also one more example I was just thinking of, like bandages. These days, the smart bandages are being used, where, the people are wearing the bandages on top of their skin. And, those bandages, they release the drugs, when they get the stimuli, they release the drugs on time. So these are like self-hearing bandages and wearable. They also have nanosensors inbuilt in them. So yeah, there also these smart materials are being used. So these bandages, they work on like how these bandages on the skin, they actually work. They have the sensors, but they also have these smart materials embedded into them, which can actually sense the temperature of the body. So it's like a temperature sensitive nanomaterial, and it can release the antibiotic for certain infection on site. So this is the kind of technology, in the terms of smart nanomaterials which is being made. So one of the smart nanomaterials even I have worked on. So I'll move on. So this is one thing which people are exploring these days, which is like nanoparticle-based theranostics. What is theranostics? It is when nanoparticles combines a drug delivery and a real-time imaging, and both are done simultaneously in a single platform. That is actually a theranostics. So, the iron oxide nanoparticles, which is they can be used for MRI contrast agents, hypothermia. The gold nanorods, they can be used for photothermal therapy and imaging. So these are the areas where people are trying to create a platform or create a system where the therapy and the diagnosis both can be done side by side. So what happens here, this nanoparticles, they function in a dual manner, and also they are more targeted and more effective. But there are benefits to it, that we can go for personalized medicine approach. We can monitor the drug release. We can do the tumor response simultaneously. But now it's the time to actually talk about what are the challenges which people are actually facing. We're talking so many things about the nanoparticles, but in terms of when we talk about cancer, when we talk about the chemotherapy, when we talk about the therapy, very less, only some of the particles which are actually being researched in our area, only some of them are making to the market. So there are big challenges. There are the bottlenecks which is actually preventing the clinical translation. The first challenge which we see is that inefficient deep tumor targeting. So if you are aware of like cancer system, it's okay, but I'll just give a brief explanation of how cancer system is. Suppose you can just imagine that cancer is a bulk of cells where the core is very, very thick and it doesn't allow anything to penetrate inside, whereas the structure, the outside cells, they are little loose and they also have got blood vessels. So what happens when we actually give the medicine inside and it goes to the cancer area, it actually has the effect on the periphery, but it does not go inside, because when the particle size is bigger than 150 or 100 nanometer, what happens is that these particles cannot penetrate deep inside the tumor. So they will only kill the cells which are outside the tumor, which is not going to give us any effect, much of the effect, because they will again grow and the tumor will again grow. But if the particles are very small, suppose we say that this diffusion inside the tumor is actually owing to the particle size. If the particle size is very small, they might go inside the tumor. But now we don't have enough particles to actually go inside the tumor, because much of them will go anywhere inside the body and they will also be cleared very fast from the body. So it's kind of a paradox where the large particles, they penetrate less into the tumors, and the smaller particles can penetrate deeply, but they have very poor retention. They cannot be retained in the tumor for a long time, and henceforth they do not show that desired effect. So this paradox, this is a bottleneck where we have to cater this problem so that we can understand and we can actually work on this problem and make the particle or the systems in such a way where this we can overcome this problem. So and another problem is the opsonization problem. Whatever goes inside the body, whatever we inject inside the body, we eat, no matter how much non-toxic it is, and we have checked the bioavailability, we have checked so many things, and still it doesn't work. Why? Because it's not just the toxicity of the material. It's that whenever it goes inside the body, there is opsonization, there is an immune clearance which happens, because no matter what you do, it is considered as a foreign body. Suppose I say that, suppose I take a cell from your body and engineer it and again inject it inside the body, still, even if it is having the properties of your own body cells, still it is counted as a foreign element. Why? Because we have engineered it. There are tweaking which we do. So there are things, the checks and balances which we have to keep in mind, and we have to keep the opsonization and everything in the mind, that when we prepare these particles. So in 2017 there was a paper in Nature where Dr. Warren Chan, they did this research where they analyzed that till now in past 2 decades, whatever research has been done in nanoparticle area, only 0.0014% of the nanoparticles, they actually reach the cancer area. 0.7% reach till the tumor periphery, but only less of them, 0.0014% reaches to the cancer area. Now you can imagine why these are not getting into the market, because basically nothing is reaching to the cancer site. How are they going to show the effect? So that's the reason, that is where the bottleneck is, and we have to work on that. So one of the rational designs which were proposed was, the stimuli responsive multi-stage drug delivery platform. So when we talk about multi-stage drug delivery platforms, that means that, there is um, a bigger particle which can go to the tumor periphery. Now we give it a stimulus, it breaks and it releases the smaller particle at that point, and then the smaller can go inside. So now here the smaller will have the longer tumor retention also, and the bigger particle which were unable to go deep inside the tumor, they will also release it, and they don't have to go deep inside the tumor. They just have to release the smaller things from inside, the active agents from inside. So this was one rational design which people started discussing, and at that point of time, even I was shaping up my PhD project. So we moved on with that thing. Also, nanomaterials are being used for photothermal therapy and photoacoustic imaging. So what photothermal therapy is, we have to understand that there are several kinds of therapies which are done for the cancer, and one of them is photothermal therapy. So photothermal therapy, it involves, yeah. So photothermal therapy actually involves using the light, typically in the NIR range, which is the near infrared range, which is more than 700 nanometer. And what happens is, when the light energy is actually absorbed by the photothermal agents, there are several nanoparticles, there are several molecules, which when absorb the light, like gold nanoparticles, they convert it into heat. So suppose we have injected something inside the body, and it goes to a particular area, and at that particular area we irradiate the laser, and there will be a localized increase in the heat, and henceforth we can the cells are killed there. So there are two things which we have to keep in mind. Not any light can be used. Why NIR light? NIR light, because the near infrared light basically penetrates deeper into our tissues. The visible light which we see doesn't penetrate much. So if something is inside, we cannot give the light stimulus to that thing. Also, the another thing is that this rise in temperature, since it is localized, it causes, there is something known as apoptosis. It actually, there is a range of temperature around 45 degrees Celsius, which makes the cells undergo a very controlled death, and henceforth killing that particular area. And side by side, these nanoparticles, which can be used for photothermal therapy, if they can be also used for photoacoustic imaging. So there is imaging. Suppose you just imagine that you have injected something in somebody's body, and you can actually image it where that particular thing is going inside the body in the real time. So that can be done using the photoacoustic imaging. How that can be done? I'll just go through it. So photoacoustic imaging, it is a technique which involves this attribute of a particular molecule, that when you irradiate the light on it, it actually generates heat. So you know that this gentleman, he is Graham Bell. He is the one who in 1880, he gave the phenomena that whenever you irradiate a light on a particular object, it actually sound waves are made. In a layman term, if you want to understand, whenever you irradiate the light get converted into sound waves. The photo gets converted into acoustic. Acoustic is what we call for the sound waves. So a photo gets converted to acoustic. So this particular principle is now being explored for the development of photoacoustic imaging. So in the photoacoustic imaging, these nanoparticles, which actually absorb the light and generate the heat, they are known as chromophores. So these chromophores, they absorb the light, the NIR light, and there is a temperature rise, and then there is a thermoelastic expansion which happens, leading to the acoustic generation, or you can say the ultrasound detection, which we can use. So everybody has seen the ultrasound machine, where you can just put it on the skin and you can just rotate it and you will get the signals. Same way here, the output signal, the kind of diagnosis you will see a clinician doing will be like he'll be doing with using the transducer on the skin, because the output which we are getting here is in ultrasound. But in ultrasound, the input also, when what goes inside the body is also ultrasound. Here, what goes inside the body is the light, and what comes, the information which we get from the body in form of which will be converted into imaging, is actually ultrasound. So this is the phenomena. So here there are multiple nanoparticles which actually can be used. One of them is gold, which has been explored, and there are several small molecules like ICG, indocyanine green, which are being used for these purposes. So for my problem statement, so these are the particles which I have just mentioned. So typically, what are the key features these particles should have? They should be 20 to 150 nanometers in age. They should have high photothermal conversion efficiency. What is photothermal conversion efficiency? How much they can actually convert the laser light which we are giving into the temperature. I know this must be getting a little difficult to understand for you, but you can definitely keep all these things in your questions and I will answer all of them. So these are the properties they should have, and they should have a strong absorption in the NIR range. Above 700, they should have a strong absorption. So these are so for my work, I used the nanoparticle, which is copper sulfide nanoparticles. So these nanoparticles are basically they are p-type semiconductors with a direct band gap. So also these particles they have a they exist in various stoichiometries ranging from CO2s so CuS2. So what happens that all these stoichiometries, they basically do not have that absorption capability above 700. What we want is a particular stoichiometry, which is the CuS form, which is the covalite form. So even when we synthesize these particles, we have to make sure that whatever particles we get in the end are the pure form of CuS, the covalite phase. So these covalite phase, people have worked on this phase for in last several years, and the photothermal efficiency is amazing. Also, one more thing is that the photothermal efficiency of this particle is based on the concentration. As you increase the concentration of these particles, the photothermal efficiency might increase. But when we talk about other nanoparticles like gold and all, the size of the particle has to increase in order to increase the photothermal efficiency. Also, the concentration also matters, but the size plays a very important role. Here in copper sulfide, even if it is of 5 nanometer, it will have a very good photothermal efficiency. So that is one advantage of this nanoparticles, because now, as I discussed, it can go deep inside the tumor also, and we can get the diagnosis of a deep tumor, and we can do the therapy of the deep tumor states as well. So it has got a strong NIR absorption ability, and then it has got a broad absorption from 700 to 1500 nanometer. We can fine tune these properties by changing the shape, structure, by changing the synthesis protocols, and many other things. Also, they are very much cost effective, very cheap, and you can easily prepare them even in your laboratories, and also scale up in the industries. Then they have the ability to do photothermal therapy and the photoacoustic imaging both. So coming on, you see these two figures. These are like two works of my which I did in my PhD, where two kinds of copper sulfide based particles we prepared. So one is this copper sulfide, where the gold was also integrated with it, the copper sulfide and the gold. And using that particle, we actually give the ROS-mediated cell like cytotoxicity along with the photothermal therapy, and side by side the photoacoustic imaging. So this paper is available. You can definitely go to Google Scholar and reach out to this paper. There's another work where we use the mucin. We have mucilage in our body. You must have seen, when you cough, there is a cough and there is a, so there also you see that this mucin is there, intestine. The mucus plays a very important role in our body. So we thought, why not take the mucin and, prepare the copper sulfide particles in a way. So we achieved very beautiful of 10 nanometer particles, and which was able to give like photoacoustic imaging, photothermal therapy. And even there is a phenomenon known as, hypoxic area. Hypoxic areas are in the tumor where there are no blood vessels, very less oxygen, nothing can penetrate there. It's very difficult to treat those areas. So these particles were even able to go to those hypoxic areas, and we can image those areas and treat those areas. So some of the results from this paper is like, these are the spheroids which we can see. These are the spheroids, like 150 micron spheroid. What exactly a spheroid is, when we take one cell and we try to multiply it in a certain way that it forms a globular structure or 3D structure, which is around of some certain size, but that depends upon your experiment. And it kind of tries to mimic how the cancer might be inside the body if the cells are all clumped up together. So you can see that these are all cells which are all clumped up together. These colors come due to different dyes. The green comes from the phalloidin, that is all the alive cells. The DAPI is written here, which comes from the nucleus of the cells. And this is our particle. So our particle, the rhodamine was attached on it for the imaging purpose and for the targeting purpose. So you can see that these particles can go deep inside this tumor. Also, when we after looking at its property to go inside this spheroid very well, even in 12 hours, then we went ahead. We want to see, how much it can affect in how much the photothermal ability is affecting in destroying these spheroids. So you can see that when there was no laser, the structural integrity was completely intact. When we just treated with our particles, nothing happens because particles are quite non-toxic. And when we just give the laser, nothing happened. We just give the particles, nothing will happen. But as we start giving the particles along with the laser, you see the integrity is lost, and they started to dissociate. So it was giving a very good photothermal therapy results. Moving on, this is the photoacoustic setup I'm talking about. So this is the photoacoustic setup. So we did the experiment on mouse, and we keep this mouse inside this photoacoustic imaging setup. And the green color, you can see these are the particles. So you see this outline here. This is actually the tumor area. This is the tumor of the mouse. So you can see that this green color is actually coming from our particles, which were acting as a contrast agent. And it was actually coming from the hypoxic area. How do we know the hypoxic area? This blue color you see, it is only Hb, that is hemoglobin. It is devoid of oxygen. So this signal is coming very prominently from this area, which means that this is a hypoxic region, and our particles were actually reaching the hypoxic region, and we got several results. So this was the photoacoustic imagings. So how we did that? First, we saw that, okay, the particles are reaching the cancer, like tumor area. Fine. Now we can give the laser and we can do the therapy as well. So then we moved on with doing the therapy. This kind of setup was used. If you see from the IR camera, there's always a temperature increase, which you can see on the surface. And this is how the tumor actually starts to burn. This is the apoptosis. The cells start to get killed. But the best part is that, the normal cells, the normal body of the mice, obviously the mice will have pain because something is burning, but the normal body of the mice will remain, the cells will be healthy, the body weight was maintained, and all the other vitals were also fine. So yes, we did achieve a very good results in terms of photothermal therapy. Now, when we talk that, I also talk this stimuli responsive combination therapy. One more work was done in our lab where what we did, we used the copper sulfide nanoparticles along with a FDA-approved targeting drug, FDA-approved EGFR targeting drug, and we incorporated inside a stimuli-responsive carrier. This was a thermo-responsive carrier. And after incorporating it, it was wrapped up with the cell membrane. Why? Because to avoid the opsonization, because so that the body does not count it as that foreign. So people generally use PEG, but PEG also has now started to show a long-term toxicity. So this was the cell membrane which was wrapped. So this was the complete composite which was created. And when we irradiated the laser on top of that, the particles, the smaller particles went inside, the copper sulfide went inside, and the drug also worked on it. And this is how, the entire system worked. So this is one result which we see. So before this entire process, before this therapy and imaging, the tumor actually looked like this. And after that it reduced to this much. And if you see in terms of, how the visualization, you can actually see the composite, the tumor, when you compare it with just the saline, the control, the tumor actually reached a very smaller size. So there was a decrease in tumor. So yes, this I'll go ahead with the conclusion and the future perspective. So you might have understood by now that there are hundreds of challenges which we face, but it's very important, for the clinical translation, because why do we do research? It's important to look from the industry perspective. It's important to look from the clinical perspective. It's important to look what actually doctors, they actually want. And then we should create our project in a certain manner. So it's important that we address the regulatory challenges and other other challenges in the clinical translation, and then move on with the project. Another thing is that whenever we are doing the the bench experiments where we are characterizing the nanomaterials, or we are doing the in vitro experiments, they should be very much extensive. Even in the preclinical studies, it should be very much extensive so that we cater all the bottlenecks of that particular field. We should try to do that. Sometimes the technologies are not available, but we should try to collaborate but the important thing is those questions should come in our mind. We should not just do for the sake of publications, although that is also important, but we should try to keep in mind that doing something with the hardcore science and to tackle the bottleneck, that is important. And also developing the standardized protocol so that if in case that something goes to the industry, you can manufacture it. So it's very important that when we prepare the particles, the steps should not be that complicated, and not many checks and balances should be there. The simpler it is, it should be good. But even if it's a little more complicated, that will be okay. So industrial aspects should be considered. So yeah, these are the things, and in the future perspective, we can keep doing the nanoparticle research and moving along in all the areas where it can be applied. So thank you so much for all your patience, and now I am ready for any questions if you have.
Hello, good evening, ma'am.
Good evening, sir.
Okay. Thank you very much for your nice presentation. So I just want to ask one question.
Yes.
Like nanomaterial can be used in many applications, as you said in your presentation, and also last day we discussed. So there are some specific criteria that which type of nanomaterial or which metal nanomaterial or any what is the criteria to use in a particular application? What kind of nanomaterial can be used in a particular application? There isn't any criteria for that.
So what I feel that for every the answer is actually hidden in your question itself. So when you say that there is a particular application, so if the application it requires that something should have a very good absorption, something should show color, or something should have a very good electrical conductivity. So we suppose I want that something should show color. The information in form of a color or a spectrometric, we can go for the gold nanoparticle because the color variation in the gold is pretty prominent. Suppose we want to go for a electrical purpose or something like that, we can go for the semiconductor nanoparticles or the conducting nanoparticles. So it definitely depends upon what kind of work you want. If you talk in terms of biomedical, first of all, it is very much important to know that what are the naturally available essential elements inside the body. If we can can make the particles out of those natural essential elements, and then it will be good for the body to eliminate it over the period of time, rather than just introducing some non-essential elements. So that's one possibility we can, work on.
Okay, got your point. Thank you very much.
Yeah, thanks.
Hello. Hello.
Yeah. Hello, sir.
Good evening, ma'am. The session was
Good
very
evening.
informative.
Yeah, thank
Ma'am,
you.
I have a question that what strategies are being developed to overcome the scalability, means
Mhm.
cost challenges of nanomaterial production for medical use?
Yeah, so that's what in the end of the slide I just talked about, that it is very important that when we sometimes, even I have done that when we prepare a project or when we write a paper, sometimes the complications in preparing those particles, it looks so beautiful to us that wow, it's so complicated, it looks so good. But actually, when you talk about the industry perspective, that's actually rejected outrightly. They don't like it. The simple it will be good. There might be several places where the complexity is actually required, and there is no other way to tackle it. Then it can be tackled. But the simplicity of the synthesis process, this is a very important property which your particles should have. Also, when we do the characterizations for a particular property, it's important that you do two characterization, three characterization, four characterizations, different characterizations, so that whenever you come to a conclusive point and you go to an industry and you tell them, okay, my thing is showing this, then you should be very much sure that this has to be this particle is actually showing that particular thing, and we have tested it completely. So these are the things which we can actually do in our preclinical setup, in our bench experiment, which gives us more probability to convert it into industrial object, or take it further for clinical purposes.
Thank you, ma'am. Thank you.
Thank you, sir.
Ma'am, can I have another question?
Yes, sure.
Yeah. Ma'am, how we can ensure the biocompatibility and safety of nanomaterials in real-time biomedical applications?
Like when you say real-time biomedical applications, what do you exactly want to ask?
Ma'am, means when we are testing these materials on the live subjects, then how we can ensure that they are biocompatible?
The first thing is that when we prepare these particles in the laboratory, generally what happens, we just do 2 or 3 tests in vitro, taking some cells, and we just do them. Actually, that much is not efficient to cater the problem. What exactly happens when we inject those things inside a living object, like a mice? So when we do, what we can do is that the kind of experiment, simple biocompatibility experiments we just run the MTT, or we do some other tests we do. What we can do, we can run those tests on several cell lines, like whatever cell lines are present inside the body, apart from the particular disease we are, targeting. Suppose we're targeting cancer, so we only see the toxicity on the cancer cells. No, we should also see that on the fibroblasts. We should see that on the macrophages. We should see that whether the macrophages are, how they are acting when they are coming in contact with the particles, because the macrophages are again present in the live animals where you are going to inject these particles. So the more rigorous experiments and more thoughtful experiments we are going to make in the preclinical setting, the better result and coherent result it will show in the animals. Also, the immune responses should also be checked. The kind of immune responses, if you are able to do, you should also do if there is any antibody response or something is coming. Try to there is a new type of what do you say, concept where people we culture several cells together, and then we treat them with the nanoparticles, and then we see that when all the cells are communicating with each other, how our particles are actually showing the results. So these are the things. The only thing I can say is that the more you will think about the body, that what are the things which actually comes into play. The blood is there, so okay, let's take the blood and check all the profiles of the blood. What is happening when it is coming in contact with the nanoparticle? Okay, this is coming in contact with the cells, so many cells. So cells, okay, what all things we we can do. Like I showed the experiment of spheroid. You can do on 2D, you can do on 3D. So you just have to make sure before going into the animal or the bigger object that whatever you are showing is actually it actually is a promising result which is coming out, not just just 1 or 2 results came positive and we move forward.
Thank you, ma'am. Thank you so much.
Yeah, thank you.
Ma'am, copper
Yeah.
sulfide, where copper is a highly conductivity material, so you are using
Yeah.
as a photothermal agent. So my question is that how it is being thermal property from a copper, where it is a highly conductive material?
Okay. Yeah, you are absolutely right that the copper is highly conductive, but things become different. Copper is different. Copper sulfate is different. Copper sulfide is different, right? And all of them have got different properties. Copper is in a pure form is conductive. It's very different. When you say that copper oxide, it is different. When you say copper sulfate, it is absolutely different. It will show absolutely different properties. And this is copper sulfide. So this is not, it doesn't comes under like conductor anymore. When you talk copper sulfide, it is actually a semiconductor. So here there are holes which are actually taking parts, and also there is a D band D transition which is happening. It is not just simply what happens in the conductor. It is a D transition which is happening, because of which the absorption properties keeps on changing with the different stoichiometries. So in the covellite phase, in the copper sulfide phase, CUS phase, this DD transition works in a way that the absorption comes after 700, and you can fine tune it using the different shapes of the particles. So it is not conducting anymore. I mean, it is a semiconductor. It is a p-type semiconductor.
Okay, at a nanoparticle range also it's working as a semiconductor.
Semiconductor. Yes, it is working as a semiconductor.
What is the particle size of it, ma'am?
10 nanometers. 10 nanometers.
Okay, thank you.
Ma'am, Pranam, ma'am. Ma'am, actually, it's a continuous question only. Can you hear me?
I cannot hear you. There is a lot of echo.
Okay.
Yeah. Just can you try again? Can you try to write your question? I can answer that. If you can just write in the chat box.
Madam, just now you told the particle size of copper nanoparticle around 10 nanometer.
Yes.
But how can we directly tell that this is 10 nanometers, because
copper sulfide,
as we do synthesis, the particle size will vary.
About your research.
Okay. So I think 2 people. Yes, yesterday.
Can I ask, ma'am? Can I ask my question now?
Yeah, please go ahead.
Yes, ma'am. Just now you mentioned that particle size of the copper nanoparticle was 10 nanometer. But
Yes.
As we do synthesis and HR10, we get a lot of variation in size. It cannot be up to one dimension. Like you mentioned, 10 nanometer, it can vary to some plus and minus size. So
absolutely.
So how can we directly conclude that
we do the image analysis, and so when I tell that it's 10 nanometer, that's the mean size I am telling you, but it is always plus minus 5, plus minus 10, like that. But so we draw a curve, we use the image J analysis, and we do the analysis for over 200 particles, and we draw a curve, a bell curve, where we get a mean at 10 nanometer. So there are several particles which are lesser than 10 nanometer, which are along 5 to 6, and there are several particles which are bigger than that, that is around 17, 18 nanometer. So that is there. Yes, of course.
So you are using your image software to calculate the mean size of particles.
Yes. So we are we are using that. Yeah.
Thank you, ma'am.
Somebody else was asking question. I think Udaya Suryan.
Hello,
Yes, sir.
I'm audible.
Yeah. Yes, you're audible, sir.
Have you taken the XRD up? By XRD, you can take the particle size. You can calculate the crystallite size.
You are absolutely right, sir. We can actually judge the crystallite size, but we have not done it using that. So what we did the particle size analysis using the image, and we did it
By
simple
imaging.
XRD. Yeah, by imaging, and we performed XRD just to know the purity of our phase. And we
Yes.
compared it with the JCPDS file. So we know that the peaks which we are getting is of the pure CUS phase, and no other stoichiometries are present there. And we also compared those peak with the HR-TEM. From the HR-TEM, we found out the D spacing, and that's how we correlated it with the
Yes,
the
yeah. That is
XRD
the
as well.
standard. Yes.
Yes, sir. We did
Yes.
that.
Yes. Because from XRD and TIM, there is no much variations. As per my
No, not
experience,
much
there is no much
variations.
experience. Yeah.
Yes, sir. Yes, sir.
Okay.
They should
Okay.
correlate.
You are
They should
given a good
correlate
informative
ultimately.
talk.
Yes, sir. Okay, sir.
Okay.
There is a question, Madam, under what criteria you selected copper sulfide as a nanoparticle for your research? So the biggest criteria why I did not go for a small. So there are two kinds of things which can be used for my research. That is a bigger nanoparticle like gold, which was already being worked on by many people. What happens, I wanted something which is smaller in size as well. It should be non-toxic as well. And it should, go deep inside the tumor, and as well as it should go show a very high photothermal efficiency. So when we talk about non-toxicity, copper sulfide basically breaks into copper ions and sulfate ions when it is metabolized inside the body. There are researches to show using PET and SPECT that over the period of 1 week, it actually gets eliminated from the body. And also in the as small size as 10 nanometer, it shows a very good efficiency of around 48%. So these two things were actually very important for me, the size and the photothermal efficiency. And that's what made me to converge to copper sulfide nanoparticle. People are going with graphene. Graphene, you cannot take it to this size. That is very difficult. And also graphene has got the structure of graphene is like, it's a sheet. So it's a sheet. You imagine that there is something which has to diffuse inside. So graphene gives a very large surface area. It's very difficult for it to diffuse inside. You need something small and compact which can actually go inside. Whether we can use carbon dot instead of nanoparticles. Of course, we can use carbon dot, but that depends upon the application. For my application, I wanted photoacoustic imaging and photothermal therapy. And for that purpose, the quantum dots were not working well because they were not giving absorption in the NIR range. So we have to go with copper sulfide. Any other questions?
Hello, madam. I
Hello.
think they're done with the questions.
Okay.
Okay. Thank you, madam. The session was very informative. You started from the introduction to the nanomaterials. You went on with the applications of the nanomaterials in the medical field, especially in cancer diagnosis and therapy. It was really useful. Thank you, madam. Thank you, madam. It was a nice session.
Thank you so much. Thank you so much for inviting me. It was
Thank
a
you.
beautiful
Maybe it
audience.
is early morning to you right now. You took the
Yeah, I
trouble.
will now head to my institute. Yeah,
Okay,
it's early
okay.
morning. It's
So thank you.
8:45.
Okay, in your busy schedule too, you made time for us. Thank you,
No,
madam.
absolutely, anytime. Thank you so much. Thank you so much,
Thank you, madam.
everyone.
Okay,
Thank you.
bye. Next session will start at 7:30. You have 10 minutes of break. So I
Good
hope all
evening
the
to
participants are in. Okay. Good evening to the participants once again. We'll be starting with our second
Evening, madam.
session today. It is by Dr. Nitin Eskale, who had given one session yesterday too. So he's not new for us, but still I'll introduce him once again. When I did the introduction yesterday, sir wasn't there. So let me do the introduction once again with him. Okay. Dr. Nitin Eskale obtained his MTech from VNIT Nagpur in 1999, PhD from the Electrical Engineering in the field of nanomaterials at IIT Bombay in 2007. During 2001 to 2007, he worked on nanomaterial cantilevers with materials coatings to carry out the research project. He is currently working as R&D head, Nanosniff Technologies, Bombay. His interested areas are nanomaterials, MEMS, material characterization of biosensors. He has over 50 publications in international journals and conference proceedings and has granted around 10 patents. His today's topic is microsensors and instrumentation for detection of explosives and gases. Over to you, sir.
Okay, yeah, thank you so much, madam, for your kind words of introduction.
Thank you, sir.
So what I will do is I will share my screen, and so today's talk is more of a work that Nanosniff has done from a company perspective in terms of what products we have developed and how they have been developed. So I hope that will also be interesting for all the participants. Okay. Yeah, so please tell me if the full screen is visible.
It's visible, sir. It's visible.
Okay,
yes, sir.
Great. So again, so as you all know that we work in the area of detecting explosives and detecting gases, and we have certain microsensors and instrumentation for doing that. So what I will do is I'll walk you through what we have done in the last couple of years and where we stand currently. So as I said yesterday, we make our own devices. We design and also fabricate our own instrumentation. We do our entire analytics of the data, signal processing, modeling, neural networks, statistics, etc. The devices of interest include cantilevers, microheaters, and microheaters with interdigitated electrodes and metal oxides. So nanosniffer is something, the explosive trace detector is something which I am going to discuss in detail today. So yeah, our work. So I think what I'm starting is with a video. I'll be playing a video which basically tells what is the need of an explosive trace detector. And I'm sure that the audio part of the video should also be visible, should also be audible to the participants. If there is any issue, I will try to explain.
Screening visitors and passengers for explosives at airports and public places has become the norm owing to increasing terrorist threats world over. Security officers routinely test luggage for explosives, anything from TNT to RTX to C4, in order to find out whether a person being screened has been in contact with explosives. Nanosniff Technologies, an IIT Bombay incubated company, has conceived, designed, developed, and manufactured Nanosniffer, a novel and low-cost trace explosives detector that exploits the physical nature of explosives in their detection. All our actions leave a trace of tiny molecules that stick to our hair, skin, and clothes, and tell a tale of where we've been and what we've been doing. During screening, the security personnel have to concentrate on sampling the area that is likely to be touched by the hands. In case of a bag, this would include handles, buckles, and zippers. Post-sampling, the swipe is inserted into the system, where a MEMS device called microheater subjects the particles to rapid heating. In case of explosives, this triggers an exothermic reaction which is picked up by the integrated temperature sensor of the microheater, thereby lending each compound its own thermal signature. The pattern recognition algorithms use these signatures to distinguish between explosives and non-explosives. Due to its affordable cost, nanosniffer can be deployed at airports, railway stations, malls, metros, corporate parks, hotels, and tourist spots. We believe that nanosniffer has the potential to be a game changer in the trace explosives detection market.
Yeah, so was the audio, did people listen to the audio?
Yes, sir.
Okay, great. So what you have seen in the audio is that what we are trying to explain is that people who have handled an explosive will have some particles that remain on their hand. And as they go around, they will be depositing those particles onto their luggage, onto their bag handles, maybe steering of their car, etc. So the job of a security person is, I mean, they are trained in that. So they have to essentially take particles out using a swab from that particular location, which is typically touched by people. And then that swab is inserted into the instrument. In certain detectors, the technology that they use is based on ion mobility spectrometry. In our case, it is based on a principle of physics, which is called as deflagration or differential scanning calorimetry. And with the help of that nano explosion that we create on the MEMS device, which is a micro heater, we are able to detect whether the thing that has been sampled is an explosive or not. So how that is done, I'll explain in the subsequent slides. Okay, so now what you see here are images of how explosives look like. So you can see that they are of different colors, they are of different particle sizes, they are of different textures, some could be liquids, some could resemble salt, some could resemble, for example, this RDX, it's similar to sugar crystals. So visibly, it is very difficult for a security person to determine whether, if a baggage has something, then that particular product is an explosive or not, or that item is an explosive or not. And therefore, people have been looking at alternative methods of verifying or checking whether the sample in front of you is an explosive or not. So that was one part which dealt with the fact that explosives have different textures, different particle sizes, colors, etc. The other well-known problem of an explosive is that it has a very low vapor pressure, which means at room temperature there simply aren't enough molecules that have evaporated from the sample so that you can smell them. So one is your sight is not helping. The second is, smelling them is also not possible because there are so few molecules. You can see here that typically the vapor pressure for most of the explosives at room temperature is of the order of -6 or even worse. So therefore, the property of your sensor has to be that it should be highly sensitive, because you are detecting something in parts per million or parts per billion. So that is the second problem. Okay, now the third problem is, you are trying to do sampling, correct? So you are trying to pick up samples. You are going to use a swab. So the swab is going to be of a certain material. Certain materials like aluminum foils, or metal strips, they can't be used. Certain plastics can't be used. Certain papers, certain cloths also can't be used. The most important property is that explosives I've already told you that it has a low vapor pressure. So they are very sticky. So the job of the security person is to pick up and we have to, sort of aid those people by providing them with a paper which is sticky for the explosives. So people have researched in this area, and we have identified a couple of materials which are very good at doing this job. So this is the paper that we are using. I will not get into the details of what material it is because that is company IP. The other problem with explosives is that many of their chemical signatures can be very similar to the compounds that we use in our daily life. For example, some of the explosives have nitro compounds, which are also present in soaps. Some of them have certain compounds which are present in the shoe polish, right? Some of them are present in the fragrances. Some of them are present in the body lotions and creams. Therefore, not only your sensor has to be highly sensitive, it has to be also highly selective. Because if you don't do that, then the security people are going to have a tremendous problem. If an explosive is detected on a traveler, then there is a certain procedure which by law, by rule, the security people have to follow, and that can cause tremendous delays. So it should be highly selective and it should be highly sensitive as well. So we have talked with a lot of security people, and when we started developing the product, we understood what their pain points are. One of the key pain point was the high percentage of false positives. So, it used to give like a positive detection for sort of every other person or every other traveler sampled, which is very, very high. Or the particular instruments which were present in the market, in fact, they are present in the market even today, they have what are called as long purge times. So let us say an instrument has detected an explosive. Then that instrument has to be cleaned, so it has to be purged. So that used to take, or it takes a long time in certain products which are there in the market. Typically half an hour, which can extend even up to 2, 3, or 4 hours. Some of the products that we have seen do not have what is called as a cold start. So there is a burn-in period, which could be as long as 24 hours. Some of the products in the market, they are swabs. They need to be baked. So you have an additional sub-product or a subsystem, which is going to bake the swabs, right? So it's also going to cost you, it's also going to cost in terms of power. So all this basically the security people told us that what it does, it slows the screening rate of the visitors and passengers, and therefore you have long queues, and that leads to irritation, that leads to unnecessary verbal exchanges. Therefore, the challenge in front of us was to sort of overcome these pain points. And we launched this product, I think, in 2021. So it took us a long time to develop it, but we have the product in the market. It has been installed at about 8 airports now. Some places in outside India have also these units. Typically, it can detect less than 10 nanograms of most of the explosives, RDX, TNT, PET, and etc. In terms of selectivity, you have like less than 2% false positives. We are able to classify the explosive with a certain, I would say, a very high probability. The analysis requires less than 10 seconds. We have practically a cold start. So within 3 minutes, the system is booted and ready for testing. We use swabs, which are highly efficient. We provide alerts which are visible and audible, both. The purge time required for us is less than 1 minute. It's all touchscreen. I'll show you that product. I'll show you how it runs, etc. So it's having a touchscreen, sunlight readable color display, etc. You can operate it at Kashmir at -5 degrees, or you can even operate it at 50 degrees in Jaisalmer in the summers. You can operate it, of course, in a covered setting, so 90-95% non-condensing humidity. You can operate it. It is backed up by a battery, and also there is a battery backup inside as well. So a main battery and a battery backup, and it weighs less than 4 kg, so it's portable. So the main value proposition is high sensitivity, selectivity, and a low deployment cost. The technology behind the instrument. So as I said, we use micro hot plates, and it has in situ temperature sensor. So what you see here is the micro hot plate device. This from here to here, the membrane is 0.5 millimeters by 0.5 millimeters. What you see are two zigzags. So the width of the zigzag is about 10 microns, and you have two such. So one acts as the heater and second acts as the in situ temperature sensor. And the thickness of the membrane is about 0.5 microns. So it's very, very thin, and it is having a very low thermal mass. So what happens is, with a very small amount of power, about 5 to 10 milliwatts, it rises from room temperature to about 500 degrees Celsius in a few milliseconds. Okay. The effect of this is, if there is a sample, if there is an explosive particle on this membrane, then it's going to deflagrate, because it's seeing a very rapid rise in temperature. And that is denoted by this. This is a differential signal. In case you don't have a explosive, let's say salt, sugar, then you will have such flat lines. So you can see that this is showing a situation which is black and white. However, life is not so easy. So there is a good amount of gray area. What we have done is, we have built in certain algorithms based on neural network, statistics, signal processing, which help us to sift through the data and determine whether the data is giving a signature of an explosive or not. So the processing is done using very high speed electronics and very intelligent algorithms. This is how the micro heater works. So you will see a micro heater getting red hot here. You would have seen earlier in the video as well. So this is how it runs. In a different light setting also we will see it. Please remember that this is a video that has been captured by a mobile phone's camera, and that too very long time back, maybe 5-6 years back or 8 years back. We had slightly different type of device at that point, but still the essence is present. The other thing was that this video was captured by driving the heater with a low frequency sine wave. And that is the reason why you can observe the variations which are very gradual. If instead of sine wave, we are going to apply a pulse, it's going to very rapidly rise to 500 degrees Celsius. So you will not be able to see the gradual change in the temperature or gradual change in the illumination. Okay, so nano sniffer. So this is the instrument. I'll be now showing a video of it. And you can see that it's a very small device. Just even the height is less than the size of a water bottle, 1 liter bottle. These are the swabs that are lying around, and it was shot in our office. So we'll go around the device, the instrument. That's the touch screen. What the operator will now do is, she will insert a swab, which is a blank swab. There is no explosive on it. And then she will run the device. So in about 10 seconds, a message will be flashed here in green color saying traces not detected. So that's the message saying traces not detected. And now you can do the next test. So again, now she is taking a swab which is having explosive on it, just 10 nanograms. And she runs it. You will see a message here now in green color saying traces detected. And there will also be a beep if you can pick it up. Yeah, so that was a beep. What I will do is I will run it once again, and this is the message in red color. I'll run it once again. Yeah, so that was the first non-explosive, and then the second. Please pick up the beep. Okay, the beep is quite faint. Anyways, I hope you could see the demo. Okay, now we were fortunate to get explosive samples from several sources like the National Security Guards or even some of the DRDO laboratories. And for doing the testing in our lab, we decided that we'll follow a certain standard, which is the ASTM standard. There is a certain designation to it. They basically say that we should be interested in certain classes of explosives, right? So, for example, nitramines, nitroesters, nitroaromatics, peroxides, etc., etc. These explosives have to be tested. Besides the blank, it has to be tested against what is called as a background challenge material for false positives. Now, an example of a background challenge material is dust or agricultural soil or even some particulate matter. In our case, we collected it from the exhaust of a truck. So, that is your control sample. Then you have to also test these explosives they're picking up from different surfaces. So it could be a metal, it could be a plastic, it could be rubber, it could be nylon and stuff like that. Okay, we did our testing at a DRDO laboratory in Pune for getting certifications as well. And so those are in place. They certified less than 10 nanograms for most of the explosives. So the way we adopted the ASTM procedure is described in this slide. So one question will be, how do you get 10 nanograms? The answer is that you have to mix the explosive in its solvent. You know how much you have taken. You have to weigh it. Let's say a milligram or so. You measure it, and then you mix it in your solvent. And then using a micropipette, you draw a small amount, right? So, and then put it again in the solvent. In this way, go on serially diluting. So you know how much you had picked up, you know how much was the quantity in the drop that you had picked up, and you know how much you have diluted. So in a couple of dilutions, one drop is very soon going to be 10 nanograms. This drop is put on the swab. The solvent is allowed to evaporate. Whatever remains is your explosive, and you have to test that. That's the procedure. So not only is the background challenge material important as far as the ASTM standard is concerned, but for field testing, so that ends the lab testing, but then you go to the field. So in the field, you are going to see a lot of other stuff. For example, perfumes, alcohols, soaps, shampoos, shaving creams, hair oil, tea, coffee, shoe polish, uh, toothpaste, tooth powder, betel nut, tobacco, lot of these things, naphthalene, camphor, and stuff like that. So we have to test 10 nanograms against these things as well. So when we pick up samples from this, the algorithm should be such that it should be able to reject these materials. How to test at 55 degrees? The reason for that is typical batteries, they have a safety temperature ambient. The rated temperature is 60. So we say, let us run our instrument at 55. Let's see what happens. That is also important from the perspective that you have to use it in a desert, Jaisalmer. So what we did is we put the instrument in a box. We put a couple of bulbs inside. We could put a couple of temperature sensors inside. We made holes in the box. It's a corrugated box. So we made holes. We stuck a couple of fans as well. We put a regulator, switched on the bulbs, attained a certain temperature, and then from the outside we inserted our swab in the instrument. So that's a very nice setup that we had made. It works. We did our field testing. So besides at a hotel in Mumbai, we also did field testing at the main gate of IIT Bombay. And I'll show a small video of how user friendly it is and how quickly it can be done, even when you are sampling a person, which is typically not done at the airports. So that is all. In 10 seconds, you get an answer. The person is cleared and you can test the next person. Yeah, so that's the message. Maybe I just show this video once again. Yeah, so, okay. Now I'll move to the next slide. So that was discussing about the product which is there in the market. Now there is something which we have been working for the last, I would say, 4 years, which is to detect gases. And this product or this project was awarded to us by Indian Oil Corporation to detect certain gases which are known to be present in a refinery environment and which are of their interest. These are examples. You could have, hydrogen, methane, H2S, ammonia, SO2, NO2, etc. So in a refinery process, detecting these gases is important because that determines what are your pollution levels as well. Again, the instruments need to be sensitive, selective, portable, because a person is going around in the refinery from one tool to the other, or from one location to the other, from one station to the other. Right? The person can always carry this portable instrument. Current solutions are there, but they are using what are called as electrochemical sensors. And electrochemical sensors tend to be large in size, consume more amount of battery power, and they also need to be frequently calibrated and frequently changed as well. So we are looking at a solid state sensor which is based on MEMS. We have done some development work on the sensor as well, which I will depict here. The way it works, so in case of explosive detection, we were trying to do a differential scanning calorimetry based technology. In this case, what we do is we are having a film, which is a metal oxide film, which is sensitive to certain gases. Now, when I say sensitive, I am saying the electrical conductivity of the metal oxide film is sensitive to those gases. Essentially, gases which are going to interact with the metal oxide film is going to have, it's going to either supply electrons or it's going to snatch electrons. So it could be either a additive, so an oxidative reaction or a reductive reaction. So based on that, there would be a change in the electrical conductivity, which is what the instrument that we developed is supposed to monitor.
ओ
And this
क्या
has to be
है
done.
1 मिनट आवाज आ रही है ना अभी?
Hello.
हेलो आवाज आ
So
रही है ना?
somebody, any issues? Hello, audience.
Sir, I think by mistake he has pressed the.
Okay, no issues. No issues.
No, sir, no.
Okay, fine. So what I was saying is that the conductivity of the metal oxide film is going to change when this gas interacts with it. So it could be either a supply of electrons or it could be a snatching away of electrons. And accordingly, the conductivity will be modulated, which is what the instrument or the electronics that you develop is supposed to monitor in real time. So this is, that is one aspect. Second aspect is that the metal oxide is more responsive to the gases at higher temperatures. In fact, in a certain window of temperatures, they are even more responsive to certain gases. And that is how we try to do the selectivity part. So at room temperature, they will take ages to respond. But at something like 250 degrees Celsius, they will respond within maybe 5 or 10 seconds. So that is the window of our interest. We want to, detect a leak as soon as possible, right? And therefore, these sensors need to be working at a high temperature. And since they need to be working at high temperature, you need to have a micro hot plate. There is no other way, or some mechanism by which they can be heated. Now, so besides the high temperature, you can also look at the way the excitation is run. Okay, you could have a square wave. You could have a square wave going to 500 or 300. You could have a pedestal kind of a waveform, so on and so forth. So that also can create different modulations for different gases with different metal oxide films. Okay. So some of the sensors that we have used are depicted here. They have come from different companies. SGX. These are all MEMS devices, by the way. One is from Renesas, the other is from Bosch, etc. Using these sensors, we have developed prototypes for gas detection. So these have been tested in the laboratory, and they are supposed to be tested in the field. So Indian Oil is going to provide us a certain refinery where this will be tested. This particular prototype is quite small. It weighs just less than half a kilo. It detects within 20 seconds and it gets initialized within 10 minutes. So from cold start is about 10 minutes for calibration. It's quite sensitive, so you can detect from 0.8 ppm to 10 ppm for NOx and SOx kind of gases. For methane, from 8% to 40% of LEL, we can detect. For H2S, from 2 ppm to about 40 ppm is what we can detect. So that's the range. The model for H2S is actually developed now. So this is a slightly older slide. The ppm accuracy might go off by about plus minus 25%. Okay. So what are the features of the prototype? So it works on a battery, and we have a charging mechanism also. In principle, it can work on line power as well. Works at high temperature, high humidity. The sensor gets recovered very fast within about 2 minutes. and you can export the data using a Windows-based software. On the screen, you have a lot of alerts, for example, for low battery or for gas detected, or if there is some internal health error, etc. Plus, for gas detected, you also have audible alerts. Again, a decent display, not touch, a substantial amount of data storage, 72 hours. So what I will now also do is I will run how this instrument works. This is a small demo which we had made again for Indian Oil Corporation, and basically it will tell you how to use it. What are the different modes? There is an auto mode and there is a manual mode, and there is a small description about data storage and retrieval. So we'll just quickly go through it. There will be a small voiceover as well. Again, this has been made in our lab by our own people, so it will not be as classy as a typical demo video goes.
Introducing our gas detection system powered by MEMS microsensor technology, developed through a collaboration between the Indian Oil and Nanostem. This innovation system offers not only gas detection but also intelligent classification and precise quantification capacities, all with added benefits of negligible maintenance requirements. Turn on the system by long pressing the on-off button. Upon boot up, the user will be greeted with three options displayed on the screen. The first option as auto sampling mode, the second option as manual sampling mode, and the third option as data transfer mode. Navigate up and down through the available option using the up and down arrow keys. Once your desired option is highlighted, press OK to make your choices. Selecting auto mode activates the system cleaning cycle, which continues for 9 minutes. Now it's on the cleaning process. Once the cleaning cycle concludes, the system will seamlessly switch to the sampling mode. Analyzing sensors.
So that's a pump that you can hear.
Promptly displays the result on the screen. Upon detecting gas, the system will classify the gas type as SO2, NO2, CH4, H2S and offer a precisely quantify the result in ppm. After the sampling for 20 seconds, the system pauses for 2 minutes, offering the convenience for configuration during the interval. Following the configurable waiting period, the system seamlessly continues its cycle till the system runs. This was about auto mode. If the user wishes to return back to a previous mode or exit from the system, a system restart will be required to facilitate this action.
So between modes, you need to do a reset.
Selecting manual sampling mode activates the sample cleaning cycle, which continues for 9 minutes. Now it's on the process of cleaning cycle. Subsequently, the user is prompted to initiate sampling for 3 cycles as it's configured by clicking the OK button. Here, the first cycle is on the process. Now, the same continues till the third cycle. As the third cycle, sampling begins.
Okay, I guess the video has stopped. Anyway, so actually what happens during data storage and retrieval as well, you need to again switch off the machine, and you have to go into, basically after restarting, you have to go in the third mode, and then using a USB drive or USB cable, you can transfer the data onto your laptop. Okay, so that is the prototype. Now to develop a prototype is sort of the end product, just before the product. But then you have to do a lot of experiments which are called as calibration experiments. You need to precisely know how much was the concentration of the gas of interest in your carrier gas. Because when you build a model, all these quantities are very important in helping to build a model, because you're going to correlate the signature of the conductivity changes with the amount of with the concentration of the gas. So this is the depiction of a calibration setup. This is also something that we ourselves build. You will see gas cylinders, you will see pumps, you will see a chamber, you will see power supply, you will see gas cylinders, gas cabinet, you will see mass flow controllers, a lot of fittings, T's, elbows, etc., and pipes. There are certain gases which, like argon, which is the first gas, which is to test how the sensor responds to inert materials. Then we try to test it in what is called as zero air or synthetic air, which is again high purity air, which is bought from from suppliers. And finally, we have the analyte. So analyte will have certain concentrations in a particular diluent gas. And based on the data, then you sort of try to develop an algorithm for the same. There are a lot of variables that we look into. For example, which gas, what concentration, what excitation profile of the heater, what is the background, what is the diluent, which sensor has been used, etc. Okay, so you have a certain experimental data. You are measuring the resistance of the metal oxide. You know what is the switch on and switch off time of the gas of interest, that is analyte. You know the temperature, pressure, humidity, and stuff like that. Basically, the health of the system. So the way the conductivity varies, this is Rmox on the y-axis on the left-hand side, and on the right-hand side you have the flow rate of the gas in ppm, in SCCM, and on the x-axis you have the time. Okay, so what we are saying is you are basically starting, and there is a certain Rmox change. At a certain instant, you are going to put in the control gas, and after it has flown, it's going to again change. Then you sort of remove, and then you're going to put in the gas flow rate for methane, and then you monitor. So this is just an example. Now this is not something which we are going to do at all times. We are going to play around with the experiments, but this is a way to depict what is happening. So for example, this tells me that at this instant I have a parameter called as Rmox start. At this point I have a parameter called as Rmox end. This is my dt. I can compute slopes, right? So that is it helps in model development or these mathematical extractions. And you could have different cases. For example, for synthetic air, for methane, you have certain response. For certain concentration, you change the concentration, you have a slightly different response, and that is what is used in development of the model. And this is an example of a very early model. Okay, so you have something Rmax, index, which we extract, etc. So please do not go into what these details are, but please try to understand that there is a mathematical model. And these are different for all 4 gases, and it also helps in classification. Okay, so these are the two prototypes that were readied, and these are sort of available for field testing now. We hope to do it very soon. Okay, so even the prototype has to be tested in a certain way, because the model that you develop for your calibration may not be exactly similar to the model that is going to be applicable for your prototype, and there are many reasons for that. So what we do is, we have the source of gas, which is a calibrated gas. So this calibrated gas is filled in what is called as a bag. It's called a Tedler bag. It will have an inlet and an outlet, and the outlet of this bag is, connected to the prototype, and then you can have suction. These are some other controls which we had done initially. So now the bag is the source. Okay, the gas in the bag is your source. For direct testing, we have also carried out such tests. You put it in a box, you have an inlet gas and you have an outlet gas, and the machine is run. So these are some of the very innovative experiments that were done for testing the prototype and for sort of modulating or playing or adjusting or fine-tuning of the model. Okay, now at NanoSniff, we don't want to depend upon these large companies such as SGX or maybe Renesas or Bosch, etc. We want to develop our own sensors, and this is the effort in that direction. So we have, as I had shown, we have our own microheater device. On the microheater, what we have done is we have put interdigitated electrodes. You can see these horizontal electrode lines. And on the electrodes, we have deposited a metal oxide film, right? So this that you see, which is a smaller rectangle, which is slightly darker, that is the metal oxide. Now, the good thing is that we have a scalable process to do it. So everything is in our control. The mask design, the process, this can be done on a 4-inch wafer, extendable to even larger wafers. There are some bottlenecks yet. We need to actually optimize our metal oxide. Those we hope to do in a different project. Okay, so these were two key things on which we had worked during, which are important from the perspective of large market products. But besides that, we also are engaged with colleges, we are engaged with laboratories, we are engaged with DRDO, we are engaged with some other institutes as well. So for DRDO, for example, we had developed a nanoliter auto dispenser. So I'll show you what their requirement was. So the requirement was that, they have a stage on which they will put their substrate. Okay. They want to dispense in a continuous manner. They want to dispense droplets of what? Of some liquid, or maybe nanoparticles in some solution. Okay, but at the time of dispensing the droplet, they also want to see it. So you have a microscope. There is as I said, there is a micropipette. They want to automate it, so you have a sort of a I won't say but it's called a linear. It's a LVDT kind of a device, which essentially pushes this component or the plunger of the micropipette. Okay. This can be positioned up and down. This can also move up and down. There is a XY stage that can also move in the right or in the forward and backward direction. And then there is a pretty heavy base plate. So this was developed for a DRDO organization. And you can see, how it works. So this is that micropipette, okay? This is again a small video. This is the microscope. and I hope you'll be able. So the entire thing, whatever microscope is seeing, is also projected on a screen. So that integration has also been done. I'll play it. So you can see the XY stage moving. You can see the movement of the micropipette. You can see the movement of the plunger now happening. It comes back. Okay. So this was fine. Then, okay, I will now show the XY movement. So that was for the movement of the plunger. You can now see the XY movement. You can see it. So wherever you want, you can actually dispense the liquid. So everything can be programmed, and you can do the display. You can do the dispensing. So very nice stepper control programs were written. They work very nicely. At the machine, we have a nice GUI. You can read the status of the instrument. You can control the camera. You can control the stage, the displacements. You need to know where your stage is, and therefore there is a reference. You can check it. You can control the pipette. You can dispense whenever you want. Okay. Now please give me a minute. I'll have some water. So there was a product called as Omnicant, which we had provided to several universities and research laboratories. They wanted to do what are called as headspace analysis experiments. So as I discussed yesterday, I had just introduced cantilevers, and I told that if a mass adsorb on it, the resonant frequency changes. If endothermic or exothermic change reaction happens, the temperature changes, so it can bend up and down if it's a bimaterial cantilever. And in case of surface stress, if you have an antibody on the cantilever and it is, let's say, specific to a particular antigen, then because of the antigen antibody binding, there will be a change in surface stress, and that will result into the bending of the cantilever. So these were the three things basically that I had explained. Omnicant deals with that. So in Omnicant, we have these micro cantilevers, 4 of them. They can be coated with a chemical which is sensitive to certain analyte of your interest. There is a piezoresistive element which is inside the cantilever. It is embedded inside the cantilever, and your electronics is monitoring the resistance of that element. The idea is if the cantilever bends, the structure gets strained, and because of the strain, the piezoresistance, its resistance will change, which your electronics picks up. I talked about compressive stress, tensile stress, so I'll not get into it again. I talked about bimetallic effect as well, change in temperature, different temperature coefficients of expansion, giving rise to a bending. Frequency change also I talked about. I also talked about optical method. You shine a laser, you pick up the reflected light using a photodiode array, or you have the piezoresistive element. You have certain contacts made with the element, let's say here as depicted here, and then you monitor how the resistance is changing. So we being electrical engineers, we have preferred the electrical technique, piezoresistive. In Omnicant, what we do is that We use a vial in which we put the analyte of interest, and at room temperature there would be some volatile organic compound, some vapor, which is going to gather in this space, which is called as the headspace. The job of the Omnicant fluidics is to take these vapors and deliver them to the sensor. So this is that instrument. What it has is, I'll describe. So there is a heater here. So from the beneath you can heat it. This is the vial in which you have your analyte of interest. You have an inlet and you have an outlet. So the carrier gas comes in, takes away the vapors to this place. This is the reaction chamber, which is expanded and shown here. There is a mass flow controller, and you will have a carrier gas cylinder outside. So the electronics which controls this and monitors the resistance of the cantilever is also inside. You see whatever is there on the screen. You can play with the keypad here. The fitment is like this, that the electronics mates in this fashion and the mechanicals mate in this fashion. You have an O-ring, so nothing leaks out. This is the cantilever which is present on the PCB, and this is the same connector. These are the same connectors. So considerable amount of control you have. You can control the temperature to up to 0.1 degrees Celsius. You can change the flow rate of the carrier gas within 0.1 SCCM in a range of 0 to 100 SCCM. So it's pretty standalone. You can download the data after your experiment. This is the very first measurement that we did. Remember, we are monitoring the resistance of the cantilever, which is a piezoresistive element. So you can see that as the gas flow rate changes, there is a change in the resistance. So the cantilever is in a way acting as a flow rate sensor. Okay. Second, the volatile organic compound is acetone. The moment it is introduced, the resistance changes. The moment you remove it, again the resistance changes. And this is repeatable. Since this was the first measurement, we did not have a control over the drift. You can see that it is drifting, but the difference is visible. And in next version, we also removed the drift. But I like to show this graph because it was the first measurement and it was successful the very first time we did it, and therefore it's very important for us. You can do a lot of experiments with such instruments. You can change the coating on the cantilever. You can change the compound, volatile compound. You can change the temperature of the different cells. You can change the flow rate of the carrier, and therefore you can do hundreds and thousands of experiments. The electronics that monitors the resistance of the cantilever is very, very sensitive. So it's like 10 ppm. With a small current of 100 nanoamperes, you can detect less than 1 ohms in a standard resistor of 47K. So you can detect something even less than 100 nanovolts. Okay. I was requested by Professor Udara that we should do something on the bio side, and therefore I've included these slides, which indicates how we can use it for biomolecule detection. So the biomolecule that we detected was or is released after a person has a heart attack, and we had worked in this area for quite some time. These days we are not working on it because of certain problem in the microfluidics, not the cantilever part, not the sensor part, but the packaging part. So that is not fully solved. Cardiac diseases are a problem in India. And they are growing for decades now. It's affecting the young people as well. It's very difficult for the doctor to say whether the person is undergoing a heart attack, because the typical symptoms which are manifested could be confused with signals like gas, some gastric problem. It's very important to detect it early, that is within the golden hour, so that if it is detected early and medicated early, treated early, the damage to the muscle, the heart muscle, is substantially reduced. However, a good amount of time actually is lost in collecting the sample, taking it to the pathology lab, getting the report back, etc., etc. Therefore, there was a requirement for a point of care diagnostic instrument where the patient's blood is sampled and very quickly tested. So our effort was essentially in that direction. These are some of the cardiac markers: myoglobin, troponin, heart fatty acid binding protein as well. They are released at different times and they peak in the blood at different times as well. The biology part is something like this. You can concentrate on this line. So this is the antibody structure. It is a Y-shaped thing. This part is common for all antibodies. Only this part is variable. And this is very specific to the antigen of interest, its shape particularly. What happens is that, let's say you are interested in a particular biomolecule. So you have the antibody to that biomolecule, okay, and that antibody is coated on the microcantilever. Now, coated means fixed. So I'll come to the fixing part. But in the presence of that biomolecule, and only that biomolecule generally, the attachment of the biomolecule will take place with the antibody. So antigen-antibody interaction happens. It's highly specific. It changes the surface energy of the cantilever. The cantilever is very thin. So the surface energy change results into a surface stress change, and the cantilever can bend up or down. So it is not due to gravity, it is due to surface energy change. But bending up and down is going to change the strain, and the piezoresistive cantilever is going to respond to that. And your electronics is supposed to pick up. So that's a long chain. But even before that, let us see how the attachment happens. So you have the support. You use a molecule like an aminosilane, aptase, for example. You attach it to the support, that is your substrate. The process is normally called as silanization. Then you attach a linker molecule, let's say glutarialdehyde. Then you attach the antibody of your interest. Okay, so that completes the bio change. And then your instrument is, or your sensor is ready for detection. We also did some what are called as optical measurements or optical studies, whether the binding is happening actually or not. So you have the cantilever, you have antibody coated on it, you put the antigen on it, and then you put a fluorescent tagged antibody on it. And you see if it fluoresces under a fluorescence microscope. Right? So if that is happening, then your method is correct. And from optical, you can move to the electrical method as well. So we did that. These were some of the optical measurements which my friends had done for certain antibody-antigen interactions called as the, this was, I think, yeah, anti-HIgG in goat, and this was for myoglobin. So these were optical measurements. Our focus was on electrical measurements. So this is the setup. The electronics is monitoring the resistance of the cantilever. These are the cantilevers coated. This is a T, which is shown here. This is the PCB on which the device has been electrically connected, mechanically connected, and fluidically connected as well. The instrument is interfaced with the PCB. This is the sensing area. You have a PBS solution, which is your control solution that comes in. It stabilizes the cantilever, and after a while you send in the antigen containing solution. Okay? And then what should happen? The resistance should change, which is what we always see. Always we see this. You enter, you put the anti-HIgG instrument or the solution here. It takes some time to travel through the tubes that I have shown, and after some time you will see that the resistance of the cantilever changes. So again, you can see that this was a big success because we are able to electrically prove that antigen-antibody interaction is happening. This was done with anti-HIgG. This was the first demonstration by any group of electrical detection of antibody antigen interaction using microcantilevers in India. Okay, now we are interested in cardiac patients, right? So we did get blood samples from a hospital in Mumbai, and we actually developed different antibodies for that. Actually, altogether different antibodies, which are called as camelets. That again is IP which we share with another party. So these camelets are highly efficient, highly selective as well. And you can see the interactions. For example, there was a normal person and a cardiac patient. You can see that in case of cardiac patient, there is always a response. For a normal person, there is no response. For cardiac patient, always a response. Okay, so that was the fun that students and researchers can have with cantilevers. But then we also have micro heaters. So what can you do with micro heaters? That is in fact even more interesting. What you can do is you have these devices, and I have given the properties, the resistance, fast rising, fast falling, very thin, low power, etc., etc. So we have built an instrument which is called as sensimer as well. That sensimer is again, at this part, instead of cantilevers, you can connect it to micro heaters as well. So when you connect it to micro heaters, you can heat the sensor or the heater so that it goes to a high temperature, let's say 500 degrees Celsius, and you can see that it cools down. You can actually record this. You can give different voltages to it. You can see the rise time, fall times. You can also generate different types of waveforms like pulse, square wave, sine wave, triangular wave, any sort of waveform that you want. Many sorts of waveforms actually you can supply to the micro heaters, and then you can do very interesting experiments. You can also add thermal mass to it, add a grain of salt and see what happens. So those are very interesting research experiments, quite teaching. So to universities and research colleges, we have a lot of material. As I said, we have micro cantilevers, micro heaters, omnicand, sensimer, etc., etc. In terms of patents and internal know-how, so there are 2 US patents, 2 Euro patents, and the topic is same. And we have Indian patents for that same thing. There is a lot of IP which is not disclosed by us. For example, how to have straight cantilevers, how to have straight micro hot plates, etc. Okay, so basically I have finished mostly my tech part. I will just have one or two slides, which is not the tech part, but which tries to explain, what we are doing at NanoStar. So this comes from a book by Donald Stokes. It talks about what is called as the Pasteur's quadrant. So Louis Pasteur, as you know, was a very famous scientist of the 19th century. We know him for a variety of reasons, for example, vaccines, but we also know him for pasteurization. He was associated with the wine industry and the beer industry of France and Germany. So there is a certain approach which Pasteur used to follow. It is depicted in this slide. So this quadrant talks about knowledge creation on the y-axis versus knowledge utilization, which is there on the x-axis. So on the first quadrant, you are having teaching oriented research, which the philosopher Sukra used to follow. It had low knowledge utilization and low knowledge creation. So new knowledge was not created, and neither was knowledge really utilized. The second case was pure applied research, which Edison used to follow. Edison, as you remember, is the bulb guy. So where utilization was very high, but new knowledge creation was very low. Third is Niels Bohr, the father of the quantum theory. That is pure basic research, where knowledge utilization is low, but knowledge creation is very high. If you look at the way he quantized stuff, and he explained the problem of the hydrogen atom, its electronic structure, etc., that is, simply tremendous amount of knowledge creation. The fourth is Pasteur, that is used inspired basic research, and that is high in knowledge creation and high in knowledge utilization. The best example is the milk curdles, right? It goes bad after a certain time. Pasteur observed this happening, tried to figure out what is happening, observed it under microscope, saw all sorts of bacteria, teeming with life, right? Curdling the milk, was very happy under the microscope. He realized that these are organic matter, and therefore they could be killed at high temperature, which is what he did, high temperature treatment, which is what the entire world does, right? So huge knowledge utilization and huge knowledge creation, because he knew what is causing the curdling of the milk. There are more examples, but I won't be taking those. I would prefer to give more times to question answers. But here what I'm trying to say is, we are looking at a path where applied goals are not inherently opposed with scientific creativity and rigor. So there is a considerable amount of creativity, considerable amount of rigorousness in the experiments that we do. So we try to choose a fundamentally new or novel phenomenon. We try to uncover the basic science underlying it, and we translate the knowledge that we gain into an application. So that is Pasteur's philosophy. And this is my last slide. So at NanoSniff, it is not that we are doing all work and we do not have fun. No, we have a lot of fun as well. If you recall, then our explosive detector, when we insert the swab, before inserting the swab, we have to press down on lever, and the lever has to come back to its place after it is released. So there has to be a spring. Now this spring has to be operated, let's say 1,000 times a day. Okay, so it has to be highly elastic. It cannot be of a plastic nature, after some time it gets deformed. No, it's not like that. So how to test it? It has to go 100,000 cycles, right? 1 lakh cycles. So you cannot have somebody doing those thing 1 lakh times. So we try to have some fun, had a toy, wrote a little bit of code, and ran it. So this is how it runs. It is left overnight. It is allowed to do 100,000 cycles, and then we see if it changes to them. So basically that is all from my side. I will give the final 15 minutes for any question and answers.
Hello.
Yeah.
Hello. Good evening, sir.
Good evening.
Sir, I have a question that apart from medical field, what are the other domains where nanoelectronics is having a great scope in the near future, as per your understanding, sir?
So medical, of course, as you mentioned, is one. So nano electronics has got to definitely do something always with either sensing or actuation. So wherever these two things have to be done, nano electronics will be present. It could be, as I said, gas sensing. It could be measurement of flows. It could be measurement of particles, the particle count, for example. So all those things are possible.
Thank you, sir.
Okay.
Any other question from the participants? Okay, I think no more questions, sir.
Yes, sir. Good evening, sir. Am I audible? Hello. Sir,
Yes.
good evening. Sir,
Good evening.
how you measure the temperature? Is a isothermal process or it is 50 to 500 degrees centigrade in a nanosniff? How you measure, sir, in that nanosniff?
Okay, so just beside the micro heater, I have another zigzag element, which is my temperature sensor. I force a current through it. I measure the voltage across it.
Okay. Like steady, assume that it is a steady state within the seconds. Am I right, sir?
Within milliseconds.
Within milliseconds.
Yes.
Okay, okay, sir. Nice.
It's very fast response.
Fast response. Okay.
And
Thank
we are
you.
doing, a measurement which is 100,000 times every second.
Okay.
Okay, so the sampling is also very, very fast.
Okay. Okay. Thank you, sir.
Yeah.
Hello, sir.
Yes.
Okay. Thank you, sir, for explaining about these different types of sensors used for the detection of gases and also explaining about the measuring instruments used. Thank you so much, sir, for your session.
So my email IDs are there with you people. Anybody wishes to contact, they can always do so for if they have any more questions. Okay. So thank you, everybody. Good night.
Thank you, sir.
Yeah.
Good night. Thank you.
Good night, sir.
Good night. Thank you.
Good night. Thank you, sir.
Good night. Thank you,
Thank
sir.
you, sir, for the wonderful session. Thank you, sir.
Thank you, sir. Thank you very much.
Thank you, sir. Thank you.
Thank you, sir. Good night.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank
Good night.
you.